Hello my dear friends, I am back again with a new video. Today we are going to discuss about the mitochondria and vacuole. Previously we were discussing about the endomembrane system. Okay, so last topic is vacuole. Before that I'll just recapitulate everything. So endomembrane system consists of four organelles: endoplasmic reticulum, Golgi apparatus, lysosomes, and then vacuoles. And we studied about endoplasmic reticulum. Okay, it is basically a system of membranes. Okay, and it can also provide membrane to other organelles at the time of cell division and also at various times. Okay, and we also discussed about its components. It is mainly made up of three components: cisternae, tubules, and vesicles. Okay, now there are further two types of endoplasmic reticulum: SER and RER, smooth endoplasmic reticulum, rough endoplasmic reticulum. We also studied the differences between them. Like RER is basically made up of cisternae and SER is basically made up of tubules. Ribosomes are present in RER, it is not present in SER. Riboforeign protein 1 and 2 are present in RER and not present in SER. Okay, for detailed description, you can watch my previous videos. RER is responsible for protein synthesis, SER is responsible for glycogen and lipid synthesis. Now let us discuss about the functions of endoplasmic reticulum. Okay, it is basically a system of membranes and provides mechanical support. It also helps in intracellular transportation because it is widely spread inside the cell, so it can be used for intracellular transportation from one place to another, like that. Okay, now RER is responsible for protein synthesis, SER is responsible for steroidal hormone synthesis and also glycogen and lipid synthesis. They are also responsible for the formation of raw materials of Golgi complex, and as well as they also form the Golgi complex. Okay, now there is cytochrome P450, okay, which is present in SER and is responsible for detoxification of toxic materials inside our body. So that's also one more function. Now next thing comes is modification. We also studied about a modification in the endoplasmic reticulum, okay, which is microsome. What exactly is microsome? Microsome is basically not found in any living cell. It is formed when we centrifuge the cell. Okay, after centrifugation we find some endoplasmic reticulums still have ribosomes attached to it. They are basically microsomes. Microsomes are used by the scientists in the in vitro protein synthesis studies. Next is Golgi complex. Golgi complex is also known as Golgi bodies, Golgi apparatus, dictyosome, idiosome, trophospongium, Mitochondria, Darton's complex, and Baker's body. It was discovered by the family Golgi in 1898. Okay, it is also known as the middleman of the cell, and also it is the director of molecular traffic inside the cell. Okay, it is made up of stack of cisternae, and cytoplasm around the Golgi complex is does not have any cell organelle present around it. It is known as the zone of exclusion. Now it has two different phases: concave phase and convex phase. Convex phase is also known as cis or forming phase, and concave phase is also known as trans or maturing phase. Now it is also made up of three types of components: cisternae, tubules, and vesicles. Now here cisternae has given there is some particular length of cisternae which is 0.5 to 1 micrometer, and there are nodes present in cisternae, okay, which is filled up with viscous fluid. Now what are the functions of Golgi complex basically? Golgi complex main function is secretion. Secretion is done in three steps. First, fusion of raw materials. Second, glycosylation, and third, finished products are then released. See, lipids and proteins are basically raw materials. So they first fuse and then goes for the second step. Their carbohydrate chains are attached to the glycolipid and are attached to the lipid and protein. And after that, they are called as glycolipid and glycoprotein. Okay, if carbohydrate
Lysosomes basically do two types of digestion. Okay, intracellular and extracellular. Intracellular are basically further of two types: heterophagy and autophagy. Okay, heterophagy basically means when lysosome digests some foreign particles. Okay, foreign particles like any food substance or any outside substance enters the cell, and then lysosome digests that thing. Then it is known as heterophagy. And when lysosomes digest the some organelle, or can say some cellular material present inside the cell, then it is known as autophagy. Okay, now extracellular digestion, its examples are fungi and osteogenesis. Okay, extracellular digestion basically occurs outside the cell. Now there is one more function, autolysis. Autolysis basically occurs when all the lysosomes inside the cell are activated, and then they digest the cell. Okay, there is one more thing, biogenesis of lysosome. Okay, biogenesis is basically where it starts from the endoplasmic reticulum. Okay, because raw materials are produced in the endoplasmic reticulum. So it goes from endoplasmic reticulum, then to Golgi complex, and then finally a lysosome is formed. So it is also known as GERL complex. G for Golgi apparatus, ER for endoplasmic reticulum, and L for lysosome. Okay, GERL complex. Now next thing comes is vacuole. Vacuole are basically either absent, or if present in animal cells, then they are very small in size. They are basically present in plant cells, and they can occupy up to 90% of the area in the cell. Okay, now vacuole further has two types. Its membrane is known as tonoplast, and inside it is known as sap. Okay, sap basically contains different types of things like water, ions, salts, excretory materials, water soluble pigments like anthocyanin or anthochlore. Okay, now there is one more type of vacuole. We have discussed about sap vacuole and gas vacuole. One more is contractile vacuole. Okay, it is basically found in amoeba. Like suppose if you keep amoeba in pure water, then water will move in. Pure means basically distilled water. Then water will move inside amoeba, and then it might burst. Okay, like if we put RBC in pure water, then endosmosis occurs and then cell bursts, but it does not happen in case of amoeba. Okay, there is contractile vacuole present in it, which in which water moves in, and then contractile vacuole moves out the water back to the area. Okay, it is basically osmoregulation only. Osmoregulation which happens in our kidney, which helps us to maintain the water and salt level in our body. Similarly, it happens in the amoeba by the help of contractile vacuole. Next thing comes is mitochondria. Mitochondria, as we all know, is also known as the powerhouse of the cell. Okay, it is also called furnace of the cell, or ATP mill, or can also say storage batteries of the cell, or semi-autonomous. Okay, so it can be called with different types of things. Whichever it is related, wherever it is related to energy, it means it is indicating mitochondria only. Okay, next comes number of mitochondria present in unicellular organisms. Minimum there can be one mitochondria. Okay, like its examples are microasterians and chlorella fusca. Yeah, it's funny names. Okay, these are basically algae. Okay, microasterians and chlorella fusca. Now maximum mitochondria present in unicellular organisms is 5 lakh. It is present in amoeba chows. It is also funny. Yes, amoeba chows contains maximum 5 lakh mitochondria inside it. Okay, now in multicellular organisms there is average about 1000 to 600 mitochondria in a cell, and maximum mitochondria is present in the flight muscle of birds. Okay, because they need to flap a lot of they need a lot of energy to flap their wings and then move in the sky. Next comes size. its diameter ranges from 0.2 micrometer to 1 micrometer. Okay, average diameter is 0.5 micrometer, and also it is of length 1 to 4 micrometer. Now you cannot see mitochondria easily in the microscope also. You first have to stain it with some color. The color used for staining it is Janus green B. Okay, after staining only you will be able to see mitochondria properly. Now let us see the structure which will help you to get it more better. See, this is the structure of the mitochondria. Okay, this is the outer membrane or outer mitochondrial membrane. It has less protein and more lipid in it. Okay, it is also semi permeable and in nature, and there are porins present inside it. Porins are basically large size channel protein. Okay, now next comes inner mitochondrial membrane. In inner mitochondrial membrane there is more amount of protein and less amount of lipid. Okay, now there are basically two types of space, outer mitochondrial and inner mitochondrial space. Outer aqueous it is also known as outer aqueous membrane or perimitochondrial space, and inner this space present inside it is known as inner aqueous membrane or matrix. Okay, now these projections finger like are basically crista, okay, or cristae. Now it has also projections inside it. Okay, these are basically oxysomes, or can also called F0 F1 particle or elementary particle. Okay, and also known as Fernandez Moran particle. When ADP and inorganic phosphate passes from these see, I have drawn one more structure, passes from this from the F1 zone, then it becomes ATP. It combines the ADP and inorganic phosphate and makes it ATP. Now this is known as oxidative phosphorylation. Okay, oxidative phosphorylation. Okay, so that's all for today. Thank you.